Hello dear students, welcome to this program. Today we are discussing light scattering and its application in biophysics. Objectives of today's lecture are introduction about light and spectroscopy, light scattering and its types, circular dichroism, optical rotatory dispersion and magnetic resonance spectroscopy. Let's first talk about light and spectroscopy. Before we hurtle headlong into the wild and woolly field of spectroscopy, we need to try to answer some seemingly simple questions such as what is light and how does it behave? These questions may seem simple to you, but they have presented some of the most difficult conceptual challenges in the long history of physics. In this century, with the creation of quantum mechanics, that we have gained a quantitative understanding of how light and atoms work. Light is an electromagnetic radiation, a phenomenon exhibiting different energies, and dependent on that energy, different molecular features can be probed. An understanding of properties of electromagnetic radiation and its interaction with the matter leads to appreciation of variety of types of spectra and consequently different spectroscopic techniques and their applications to the solution of biological problems. Spectroscopy is the study of interaction between matter and electromagnetic radiation. Typically, one can observe two distinctive classes of spectra, continuous and discrete. For a continuous spectrum, the light is composed of a wide continuous range of colors, that is, energies. With discrete spectra, one sees only bright or dark lines at very distinct and sharply defined colors. Now let's talk about light scattering. Light scattering is a form of scattering in which light in the form of propagating energy is scattered. Light scattering can be thought as the deflection of a ray from a straight path, for example, by the irregularities in the propagating medium, particles, or in the interface between two media. Deviations from the law of reflection due to the irregularities on a surface are also usually considered to be a form of scattering. The scattering of light yields a number of valuable insights into the properties of macromolecules, including the molecular mass, dimensions, and diffusion coefficients, as well as association or dissociation properties and internal dynamics. Most of scattered light possesses the same wavelength as the incident light. This phenomenon is called elastic light scattering. When the scattered light has a wavelength higher or lower than the incident light, the phenomenon is called as inelastic light scattering. Now let's talk about circular dichroism. Circular dichroism, that is CD, spectroscopy measures differences in the absorption of left-handed polarized light versus right-handed polarized light, which arises due to the structural asymmetry. The absence of regular structure results in the zero CD intensity, while an ordered structure results in spectrum which contain both positive and negative signals. Circular dichroism spectroscopy is particularly good for determining whether a protein is folded and if so, characterizing its secondary structure, tertiary structure and structural family to which it belongs. Comparing the structure of a protein obtained from different sources, example, species or expression systems, or comparing structures for different mutants of the same protein. Studying the conformational stability of protein under stress. CD is excellent for finding solvent conditions that increase the melting temperature and the reversibility of thermal unfolding conditions which generally enhance shelf life. Determining whether protein-protein or protein-ligand interactions alter the conformation of protein. If there are any conformational chains, this will result in spectrum which will differ from some of individual components. Small conformational chains have been seen, for example, upon formation of several different receptor-ligand complexes. Now let's talk about optical rotatory dispersion. Optical rotatory dispersion is the variation in the optical rotation of a substance with a change in the wavelength of light. Optical rotatory dispersion can be used to find the absolute configuration of metal complexes. Optical rotations usually are measured at just one wavelength, that is 589.3 nanometers, simply because sodium vapor lamps provide an especially convenient source of monochromatic light. Measurements at other wavelengths are less easily made without specialized instruments, with which relatively few labs are currently equipped. Nevertheless, much information has been obtained about the structure and conformation and configuration of organic compounds from measurements of optical rotation as a function of wavelength, that is, optical rotatory dispersion. Like other phenomena involving interactions between electromagnetic radiation and organic molecules, as in infrared, ultraviolet, and NMR spectroscopy, optical rotatory dispersion curves often are quite sensitive to small changes in structure. Let's give an example of rotatory dispersion curves for enantiomers of cis and trans 10-methyl-2-diclones. Only a small positive rotation is observed for particular enantiomers at the wavelength of sodium line, that is 589.3 nanometers, compared to the large both positive and negative rotations found at wavelengths between 270 nanometers and 400 nanometers. If we measure the rotation as a function of wavelength, and if as we approach shorter wavelengths, the rotation rises to a maximum before changing sign, as it does with trans isomer, then the compound is said to exhibit a positive Cotton effect. The opposite behavior, as with the cis isomer, is called negative Cotton effect. The wavelength at the center point for the very rapid change in the rotation for trans is 300 nanometers and corresponds to the absorption maximum of the carbonyl group in the ultraviolet absorption curve of the same compound. Thus, excitation of carbonyl group by absorption of ultraviolet light and strong rotatory dispersion of polarized light are associated phenomena. In fact, when a substance exhibits a Cotton effect, not only does it transmit clockwise and counterclockwise circularly polarized light with equal velocities, it also absorbs two forms of light unequally. At last, now let's talk about magnetic resonance spectroscopy. Magnetic resonance spectroscopy (MRS) is a means of non-invasive physiologic imaging of the brain that measures relative levels of various tissue metabolites. MR spectroscopy is a modality that is available on most state-of-art clinical MR scanners. For the brain in particular, MRS has been a powerful research tool and have also been proven to provide additional clinically relevant information for several disease families such as brain tumors, metabolic disorders, and systemic diseases. The most widely available MRS method, proton spectroscopy, is an FDA-approved procedure in the US that can be ordered by the clinicians for their patients if indicated. Other methods such as phosphorus-31, carbon-13, or fluorine-19 MRS have been successfully applied in humans. But with the ever-increasing importance of clinical MR imaging, these exotic and time-consuming applications have been pushed to the side and are only available at few academic centers. In addition, proton MRS does not require any additional hardware beyond what is already being used for MRI. Dear students, now let's know what can be measured with the magnetic resonance spectroscopy. MR imaging maps the distribution and interaction of water, that is, its hydrogen atoms, with tissue. In contrast, proton MRS analyzes signal of hydrogen protons attached to other molecules, whereas for MRI, only a single peak is being mapped. The output of MRS is a collection of peaks at different radio frequencies, representing proton nuclei in different chemical environments. Only small mobile chemicals with concentration of greater than 0.5 millimolar per gram tissue can be observed with in vivo MRS. This leaves most of neurotransmitters out of reach for this method. Exceptions may be glutamate, G-aminobutyric acid, that is GABA, and aspartate. In addition, large immobile macromolecules and phospholipids, myelin, proteins, RNA and DNA are rendered invisible to MRS. The network of small molecular weight amino acids, carbohydrates, fatty acids and lipids that can be measured is tightly controlled in the brain by enzymes, and all but a few key molecules, MRI invisible messengers and neurotransmitters, are kept at remarkably constant concentrations. It's for this reason that reproducible MR spectra of the brain can be obtained with robust methods are applied. In sequentially studied individual healthy controls, the single greatest variable may not be biological or diet-imposed variation, but the practical and avoidable inaccuracy of positioning of the subject, problems with the identification of previously selected region of the brain, and imperfect stability of MR hardware. The biochemical fingerprint of tissue will be abnormal when there is a structural damage, for example, trauma, tumor, degenerative diseases, etc., or altered physiological conditions, for example, interruption of blood flow, and biochemical or genetic problems. The metabolic fingerprint also varies with the brain regions studied. Some applications of MRS are: distinguish neoplastic from non-neoplastic masses, or to distinguish primary from metastatic masses, to distinguish tumor recurrence versus radiation necrosis, prognostication of disease, mark region for stereostatic biopsy, monitoring response to treatment, or it can be used as a research tool. This is all about today's topic. Hope you have understood. Thank you.